Okay, class. Now we're going to start on this document here. And we're going to do the best we can on this document. This is explain excitation, contraction, coupling. What I want you to do, though, is print this document out and lay it as you study this. Also, before you study this, I want you to extensively study my electrophysiology module. Now, you'll see that in the D2L. Study every aspect of the electrophysiology module. Then go back and review what we did on the anatomy of the skeletal muscle cell so you can see all the components. Once you do all of that, then you'll understand exactly how this goes. All right, so we're going to walk through, but again, you have to have this in your mind before you can do that. Action potentials travel down the axon terminal into the synaptic end bulb. The action potential, sodium and potassium gates, which you'll learn in the electrophysiology model, travel over the synaptic end bulb and then cause the opening of voltage-dependent gates to calcium. The calcium then will come in to here, and the calcium will cause the synaptic vesicles to pull down to the surface. The calcium will cause the synaptic vesicles to pull down to the surface. Okay? When the synaptic vesicles pull down to the surface, what ends up happening is, when they pull down to the surface, then they fuse with the surface, and by a process of exocytosis, they release the calcium. Into the synaptic cleft. See, all that's in that electrophysiology. Okay? Some of the calcium, they release the acetylcholine. So let me go again. The calcium ions come in because calcium's in a higher concentration outside the cell than inside. The calcium shoots into here in this synaptic end bulb. That causes these synaptic vesicles to pull down to the surface. These synaptic vesicles to pull down to the surface. When the synaptic vesicles, which contain a neurotransmitter substance called acetylcholine, when these pull down, they fuse with this membrane. When they fuse with the membrane, then they open up, and by exocytosis, acetylcholine comes ballooning out. Now, this is extracellular fluid. Some gets whisked away, but some is pulled across to acetylcholine receptors on the skeletal muscle membrane. I need 2 per receptor. When that occurs, that will cause the opening of ligand gates, again in this module. Ligand gates. The ligand gates are open because of the acetylcholine attaching. The ligand gates will open and allow sodium to come in and potassium to come out for reasons to be seen when you study that. More sodium comes in than potassium comes out, and so it makes this membrane have enough voltage to reach threshold. The threshold then causes action potentials to occur in voltage-dependent gates. The action potentials then travel in all directions around this membrane. So let's go back here. Acetylcholine crosses the synaptic cleft and 2 molecules attach to each receptor on the muscle. This causes the opening of a ligand-gated channel which allows sodium to flow. This results in a motor end plate potential to be studied, which is due to sodium ions entering the muscle cell and potassium ions exiting. Because of the drive for sodium to enter is greater than potassium to exit, the membrane is brought to threshold. The threshold voltage initiates action potentials that travel along the muscle cell in all directions from this motor end plate. That's this area here. The action potentials travel along the membrane and into the T-tubules, the transverse tubules we talked about, causing the opening of calcium release channels, in the terminal cisternae of the sarcoplasmic reticulum. The calcium inside the terminal cisternae is held by calsequestrin, also known as calcium binding protein. Calsequestrin has a high capacity but low affinity for calcium ions. This means it holds a lot of calcium but not that tightly. Thus, when the calcium release channels open, they're going to open right into there. When they open, the high concentration of calcium rushes out down its concentration gradient, and it's going to rush into the muscle cell cytoplasm, and that is also called the sarcoplasm. Remember sarcoplasm? Okay, sarco for muscle. Note, the sarcoplasmic reticulum is a storage site for calcium ions in skeletal muscle cells. Along the membrane of the sarcoplasmic reticulum are calcium pumps. And see, these calcium pumps constantly pump calcium ions from the sarcoplasm into the lumen, which is also called the cisternae of the sarcoplasmic reticulum. The calcium ions are pulled towards the ends, the terminal cisternae of the sarcoplasmic reticulum maze, because of the affinity of the calsequestrin, which is there, which is also called calcium binding protein. So the terminal cisternae holds a lot of calcium ions, okay, but it doesn't hold it that tightly. Now let's go further. The rushing out calcium ions go into the muscle cytoplasm, which is also called the sarcoplasm, some being pulled to troponin C. Remember that troponin I, C, and T. So troponin C is part of a troponin complex, which we mentioned, which consists of troponin I, C, and T. Troponin I attaches to actin. Troponin T attaches to tropomyosin, and troponin C binds to calcium ions when available. They are now available because of these action potentials release the calcium from the terminal cisternae. Under resting conditions, the tropomyosin filamentous molecule covers the myosin binding site on G-actin. Let's go back to that picture. See, that was this picture here where that's covering that. That was this picture here where that's covering that. So under resting conditions, the tropomyosin filamentous molecule, which is that I'm speaking of, covers the myosin binding site on G-actin. See, that's the G-actin, that's the myosin binding site. It must be uncovered for actin to be able to bind to myosin. The calcium ions give this ability, because when the calcium attaches, it's going to shift the complex and move this. So let's go further. The attaching calcium ions to troponin C causes the troponin complex to twist. That twist, put a comma there, causes it to twist, comma, save that. That twist causes tropomyosin to shift in such a manner that it allows uncovering of the myosin binding sites on the G-actin. Okay? This action makes actin ready to bind to myosin, but myosin's not ready yet. So we need to turn our discussion to myosin. Now what we've done is to remove that ribbon. Listen to me. What we've done is to remove that ribbon. See, remove that ribbon. Okay. Once that ribbon was removed, you opened up some sites there. Let's see if I see a picture of that. Okay. So now you remove that ribbon. So here is the action potentials coming down into the T-tubule. Here you're opening up these calcium release channels. The calcium shoots out and goes into the cytoplasm. Some of it attaches to troponin C, and when it attaches to troponin C, it shifts the troponin complex, shifting tropomyosin off of the myosin binding sites on actin. Okay? Note, on each myosin head, 2 per myosin molecule. We've talked about that. 2 per myosin molecule. Okay? Are 3 things, and we talked about that, which is the ATP binding site, actin binding site, and the ATPase. Okay? All right. Analogy. In order to understand the myosin action, let's envision the hammer of a gun. It takes energy to pull the hammer back, but all you have to do is squeeze the trigger and the hammer automatically flies forward with tremendous force. Okay, so here we go. In the relaxed state, the myosin head is flexed like this. So this right here would be like this. It would be flexed like that. Okay? It's flexed. The action potential is called the activation of ATPase enzyme. This ATPase enzyme causes the breakdown of ATP to ADP plus the P. Okay? The ATP plus P resultant products stay attached to the myosin head after it's all over, temporarily. Okay? The energy derived from the breakdown, catabolism ADP, is used to pull the, i.e. cock the myosin head back into an extended position, like this. The power is going to be in the recoil pulling the trigger. So in other words, it's like this. Now when that occurs, okay, when that occurs, let's go down here. So I'm going down here now. When that occurs, let's go further. Okay, and that splits down. See how we had it? And then when that ATP split, it came back, and then it's going to attach eventually. Okay, so here we are. There's the calcium. Here we are right there, going further. Okay, that's the cross bridge. But I wanted another picture if I see one. Okay, here's this thing here now. Here we are. This would be in the resting state with ATP. The enzyme splits ATP into ADP plus P, and the energy cocks this back. When that's cocked back, then you can do some attachment. But let's go. When the ATP on the myosin head is catabolized, this allows the acting binding site on myosin head, okay, to be uncovered. So when this cocks back, you can uncover that heat, the actin binding site on myosin. The two bind together. So once you do that, the two can bind together. Okay? Now, let's remember something. Okay? Two can now bind together. They're bound together, let's say. Remember, the ADP plus P is still attached to the myosin. Even though you got the energy from it, you kept this still attached. The ADP plus P finally drop off. So in other words, you cock back. When they drop off, see right now it's still attached. Okay? When they drop off, okay, when that occurs, they drop off. The myosin head attached to actin is allowed to recoil, to now pull in and slide in, and pull the actin and over the myosin, slide in. This causes shortening of the sarcomere, resulting in contraction of the muscle cell. Okay? Note, the myofibrils are located on the interior cell. They slide in. Now, we already talked about that. Now, the other thing I want to mention is this. Okay. The myofibrils located on the interior, they slide in. Okay. However, in order for the muscle cell to shorten, the myofibrils also must pull the cell membrane. Thus, there must be an attachment of that. That's where this comes. The attachment is due to this right here, which is called dystrophin. The attachment is a large protein, and it hooks these myofibrils to the muscle cell membrane. So when they come in, they pull the membrane too. That's what the problem is in muscular dystrophy. The protein is not right. Now, the last thing is to look at this. Rigor mortis. Rigor mortis occurs when you don't have ATP. You can read this. When you don't have ATP, you can't suck the calcium back. If you can't suck the calcium back, it stays attached. So I'm going to close here.